from science. They might be trying to take on science.
Hi, Dr. H here. This video is going to review the Unit 1 of AP Biology, which will cover the introduction to the course and some basic characteristics of life. We'll go over some of the introductory chemistry concepts that are important, including macromolecules, and then finish up with a very brief discussion of cells and a few of the important organelles. So in terms of introduction, the College Board has broken down the curriculum of AP Biology into 4 big ideas, and those ideas are listed here. And I will cover these throughout these review videos. Some of them, such as evolution, will have their own video. They have their own unit. Energy definitely will have its own unit. But you should be able to identify these main themes running throughout everything we covered in the class. So first off, what does it mean to be alive? There are lots of different definitions of living things. We talked about a few of them in class. But these are some basic ones. All living things show all of these characteristics here. Order, living things are very organized. All living things can evolve and show adaptation to their environment. They respond to their environment. So all of these things are in place in all living things. And we'll talk about a couple of them a little bit more in detail here, especially order. Living things are very, very complex, much more complex than any inorganic or any non-living material that we have on Earth. And one important sort of characteristic that comes out of this order, this complexity, is something that we call emergent properties, meaning that when we take small things and put them together in a certain way, we come up with brand new properties when we put them together correctly. And those new properties are what we call emergent properties. So if we look down at the smallest level here, at the atom, right? So we have all of these carbon and hydrogen and oxygen atoms, and they all have certain characteristics, but when we put them together in just the right way, we get a molecule, and that molecule has very important, unique properties that were not apparent at the atomic level. Okay, and then we take a bunch of those molecules, put them together in a specific way, and then we get an organelle. And that organelle, again, will have unique characteristics that were not apparent at the molecular level. And these emergent properties run all the way up through all of these levels of complexity, all the way up to the biosphere, which is the entirety of all living things and also all the non-living things that they interact with on the Earth. Another very important characteristic of life, which I will go into briefly here, is regulation. Okay, living things maintain homeostasis inside, whether that is their temperature, the internal salt concentration. All of these things are very important. We talked about 2 different types of feedback regulation in class. The first one here is what we call positive feedback. And this one is quite rare in biological systems, though it is still important. So here we have a biochemical pathway going from compound W, is converted to X, converted to Y, converted to Z. And this is done through the activity of 3 enzymes, enzymes 4, 5, and 6. So in positive feedback, what happens is the final product here, which would be Z, goes back and activates one of the enzymes. In this case, it is activating enzyme 5. And that leads to more product Z being made, which would then in turn lead to more activation of enzyme 5 and then more product Z being made. So more and more and more of the final product would be accumulating. So this positive feedback in terms of homeostasis, this tends to move living things away from a set point. One really good example in biological systems, childbirth, the uterine contractions. So those are controlled by a couple hormones. Those hormones stimulate the uterus to contract, and those contractions cause the release of more of those hormones, which then causes the uterus to contract even stronger, which then causes more of the release of the hormones, and so on and so on. And the other type of feedback is much more common, which is called negative feedback. And this tends to keep a cell or keep an organism closer to the set point. So here we have another biochemical pathway. This one's going from A, B, C to D. 3 enzymes again, enzymes 1, 2, and 3. But in this case, since it's negative feedback, the end product D goes back and inhibits one of the enzyme. It inhibits enzyme 1. So this stops the production. So in this sense, once the organism has enough of the final product, in this case molecule D, it stops producing that product. So it maintains that balance. It maintains that set point of having just the right amount of this molecule present. And this is much more common in biological systems. Virtually every biochemical pathway is controlled by some sort of negative feedback loop. One other thing that came up in the beginning of the course was the scientific method. And this is the basic scientific method that you may have seen previously. And I think, as I said, this was a little bit too simple for us. Throughout the year, we did a number of labs where you were designing your own experiments. Make sure to go back and look those over and be familiar with all the different parts of an experiment, right? Control group, experimental group, dependent variable, independent variable, know how to go from a table of data to a graph, what type of graph is appropriate, which axes should the data be graphed on. All of those things are very important to designing an experiment. And for the most part, we covered that very well throughout the year. So moving on to our next topic of really quick chemistry review. Not going to go into a whole lot of detail about the atomic structures, mainly because that doesn't show up all that much on the AP test, and because there's not really that many elements that occur in biological systems. These are the major ones here that show up in the human body. I see majority is oxygen and carbon, with a good bit of hydrogen, nitrogen thrown in there as well. So if we were to look at a biological periodic table, we could kind of simplify it as this. I would certainly make sure you remember what these numbers refer to. The atomic number there, that refers, of course, to the number of protons in the nucleus. And for a neutrally charged atom, that will also give you the number of electrons. And the atomic mass will give you the protons plus the neutrons, and that those are both found in the nucleus. One atomic property which becomes very important to biological system is the electronegativity of the atom. And you can kind of think of electronegativity as how strongly does the nucleus, do the protons in the nucleus, pull on the outermost shell electrons. So if the electrons are very far away, they won't be pulled quite as hard. Or if there aren't as many protons in the nucleus, they won't be able to pull quite as hard on the electrons. So the most electronegative atom that we come across in biological systems is most likely oxygen. Okay, there is a general trend as you move up and to the right on the periodic table that elements become more and more electronegative. So fluorine is a little bit more electronegative than oxygen, but as I said, we don't really see that in biological systems. So this concept of electronegativity is very important. Okay, it's how strongly does the nucleus pull on its electrons. And this comes into play when we start making chemical bonds. So first off, why do elements form chemical bonds? They want to have their outermost shell of electrons full. Okay, that is the stable state. And they will do this either by giving away electrons if they have a very low number in their outer shell, or by gaining electrons if they're very close to a full shell. So for most atoms that we talk about, that number that they're looking for is 8. So electronegativity plays into this. Basically, we look at the difference in the electronegativity of the two atoms that are forming the bond. So in this case, we have sodium, which has a very low electronegativity, and we have chlorine, which has a very high electronegativity. So there's a big difference in the electronegativities between the sodium and the chlorine. So much so that chlorine is actually able to pull that electron away from the sodium. And the sodium then becomes the positively charged sodium ion, and the chlorine, since it gains the electron, becomes the negatively charged chloride ion. So both of these atoms are now stable. Sodium has a full outer shell. It lost the one. And chlorine also has a full outer shell. It gained one, where before it had 7. And since these two ions now have opposite charge, they end up sticking to each other. And this is what we call an ionic bond. Okay, this is when two atoms of very, very different electronegativities get together and trade electrons. So what happens when the atoms are a little bit closer in electronegativities? In this case, we have two hydrogen atoms, and they will of course have the exact same electronegativities. So one atom can't really pull an electron away from the other completely. So they end up sharing, okay, and they will share the one electron between them, and that will form what we call a covalent bond. Electronegativities that are very far apart will form ionic bonds. Electronegativities that are closer together will form covalent bonds. But there's also something in the middle, where one atom is a good bit more electronegative than the other atom, but not quite enough to pull the electron away from it. And in that case, we have what's called a polar covalent bond. And a really good example of this is a water molecule. Okay, oxygen, as I said, is highly electronegative, so that pulls very strongly on the electrons. The hydrogen atoms that are on the other end of the water molecule have a very weak electronegativity, so they can't hold on to their electrons. So oxygen tends to pull the electrons up towards it, and hydrogen, though it's still sharing, kind of gives up its electrons to oxygen. So that ends up with the water molecule being partially charged on each side. The oxygen end of the molecule has a partial negative charge on it, and the hydrogen end has a partially positive charge on it. Okay, and this is what we call a polar covalent bond. Okay, the electronegativities are far enough apart where they don't share equally, but they're not quite far enough away where the oxygen can pull the electron completely away from the hydrogen. And these polar covalent bonds set up something that's very, very important, this intramolecular bond. In this case, this is called a hydrogen bond. Okay, so when the partially positive end of one polar molecule interacts with the partially negative end of another molecule. So in this case, we have a polar molecule of water forming a hydrogen bond with a polar molecule of ammonia. And these hydrogen bonds are very, very important. They are quite weak. They are much weaker than the ionic or the covalent bonds. But water is able to form so many of them that the bond strength does start to add up. And water ends up being one of the most unique chemicals on Earth. And all of its unique properties, all of these emergent properties of water are due simply to these hydrogen bonds. So this is what water looks like when it arranges itself in space. You see the negative ends of one water molecule will align themselves with the positive end of another molecule, And this is all held together by hydrogen bonds. All of the important unique properties of water are all due to the ability of water to form these hydrogen bonds. And there are a few very important emergent properties of water. I'll go over a few of these here. And again, these are all due to hydrogen bonding. Very important that you remember that. Water acts as a very, very good solvent, especially for polar or ionic molecules. And the reason for that is because it has two charges on it. Okay, so here we have a big chunk of sodium chloride salt being dumped in some water, and the sodium ions and the chloride ions come apart. They dissociate in the water, and the sodium ion there is surrounded by the water molecules with the negative ends facing in. So everybody's happy. The positive sodium gets some negative oxygen atoms to interact with. And the chloride ion, the negatively charged ion, is surrounded by water with the hydrogen, the positive hydrogens going in. Okay, and that makes everybody happy because remember, the opposites attract. So that is why water works so well to dissolve so many ionic compounds. When water freezes, it floats, right? This is due, again, to the hydrogen bonds. The reason that water floats is that when the ice forms the crystal lattice, the hydrogen bonds become stable and they become, on average, just a little bit longer. So the water molecules are a little bit further apart. Okay, when the molecules are a little bit further apart, that makes the ice less dense than the liquid water, so of course it is going to float. So why is that so important for life? That means that in the wintertime, when the lakes and the ponds freeze over, they freeze from the top down. And that top layer of ice will form an insulating layer, so the water underneath will remain liquid and will remain at a temperature which can support life. So the little shrimp here can survive the winter right underneath the top layer of ice. If solid water sank and was more dense, then everything would freeze from the bottom up, and everything would turn to solid ice every winter, and life in the water could not have survived. When water molecules stick to things, this is again due to hydrogen bonds. When water molecules stick to something else, then that is called adhesion. When water molecules stick to themselves, that is cohesion. And these two properties kind of work together to help water move up the trunks of very, very tall trees, or really any plant. Okay, so there's these really small water conducting cells in the plant, the xylem, and the water moves into the plant from the roots, and as it goes up the trunk, you can kind of visualize this as a intact chain of water molecules all held together by the hydrogen bonds. And as one water molecule leaves out the top, then it pulls the whole chain up. And that pulling force is conducted all the way down into the soil, into the water in the soil. And that is very important for moving the water up plants and up the tallest trees. And adhesion and cohesion also work together to create the surface tension. And you can see this little water bug here. You can actually kind of see the little dimples that the bug is forming where it's standing on the surface of the water. Okay, because of the adhesion and cohesion, water forms that kind of little skin on top. And all of these important properties, all these emergent properties of water are all due to the hydrogen bonding. One other very important and interesting thing that water will do is it will ionize a little bit. Okay, every now and then a hydrogen bond becomes strong enough to actually pull a hydrogen off of another water molecule. So here we have two water molecules interacting with each other through a hydrogen bond, and spontaneously one hydrogen bond, or one hydrogen, has left a water and gone to the other. Okay, so that forms two ions. The positively charged ion is the hydronium ion, which is H3O positive, sometimes also referred to just as hydrogen ion, H plus. And the negative ion, the OH, is the hydroxide ion. So this happens at a very, very low level in pure water, but it's also very important because this forms the basis of the pH scale. Okay, and the equation for pH is written there. pH is the negative log base 10 of the hydrogen ion concentration. Now, you do not need to memorize that formula. You most likely won't even need to use that formula, but you do need to understand what that formula means. So pH is a log scale. So every movement of 1 unit on the pH scale represents a 10-fold difference in the hydrogen ion concentration. So the lower numbers on the pH scale refer to more acidic compounds, and that is a higher concentration of hydrogen. So pH 1 is 10-fold more acidic than pH 2. Okay, and as you get above pH of 7, which is neutral, substances become more basic. Okay, and again, everything is based on these powers of 10. Okay, so that's the important part to remember about the pH calculation there. All right, so other chemistry we talked about, we spent a good bit of time talking about organic chemistry and biological polymers. Organic chemistry is the chemistry of carbon. And carbon is a very interesting molecule. It makes all kinds of interesting shapes. It bonds with lots of different elements. So it can form straight chains like ethane or propane. It can form branch chains like the 2-methylpropane there. It can form double bonds, as the butene isomers there show. And it can form rings like cyclohexane, benzene. So all of these really interesting shapes are possible because carbon can form these 4 covalent bonds to other elements. And it will form bonds with lots and lots of different elements. So isomers is a fairly important concept. So an isomer is 2 different compounds that have the same chemical formula. So they are made up of the same numbers of atoms. So the first type here, structural isomers. This is isomers that differ in the way those atoms are put together. Okay, so they will have the same formula, but the atoms are arranged in space a little bit differently. The second type here, geometric isomers. These deal with a carbon-carbon double bond. And one thing that comes out of a carbon-carbon double bond is the carbons cannot freely rotate around that bond. Okay, single carbon bonds, the carbons can flip all around and change shape and move around in space. But once you put that double bond there, the 2 carbons are stuck. So they can no longer rotate. If there are 2 things on the same side of the carbon-carbon double bond, that's what we call the cis isomer. Okay, if they are on opposite sides, then it is considered to be the trans isomer. Okay, so cis on the same side, trans is on the opposite side. Okay, and the last form of isomers can be a little bit hard to understand, but they do actually end up being one of the more important biologically, and these are enantiomers. Okay, and this is when a carbon atom has 4 different things attached to it. Okay, and you can attach these 4 different things in different ways, and you can come up with these stereoisomers or enantiomers. So they are non-superimposable mirror images. Okay, and your hands is a very useful way of thinking of this. Okay, your hands are mirror images of each other, but you cannot put them on top of each other and have them line up. Right, so these biological molecules do the same thing. And this is very important because for many molecules, one form will be biologically active, such as all the amino acids are all enantiomers, and one form of the amino acid is used by cells. The other form, even though it's the exact formula, is not used. Biological molecules, there are four main groups, and most of them are all what we call polymers, which are big, long chains made up of smaller subunits called monomers. Okay, and there's two important reactions here. The first one here, the dehydration reaction, that is adding a new monomer to the chain and making the polymer 1 unit longer. Okay, and it's called dehydration because during the reaction, one molecule of water comes out. Okay, so one molecule of water is formed every time a new monomer is added to the polymer. The other reaction is the exact opposite, and that is hydrolysis. In these reactions, a polymer is broken down into its monomers. And it's the exact opposite. Instead of taking a molecule of water out, a molecule of water is added in. Okay, so dehydration creates a polymer. Hydrolysis breaks that polymer down into the monomer. So the four classes of biological molecules that we talked about, I would definitely go over or definitely know what the monomers are for each of these and the major functions. Okay, I would not worry about learning details or learning specific names of any of these molecules, but if you know what is contained on this chart, that should be enough to get you through. One important note here is about lipids. Technically, they do not have a monomer polymer type structure, but they are made up of smaller bits. We have the triglycerides here are made up of one glycerol molecule attached to 3 fatty acid molecules. And the last topic that we covered in Unit 1 were cells and organelles. And in terms of cells, we talked about a few things. We talked about why cells are so small, why multicellular organisms are made up of lots and lots of small cells rather than just a few bigger cells. And the reason, of course, comes down to surface area. It's very important that the surface area be kept high so that diffusion, which we'll talk about in the next unit, can move things into the center of each cell. And then in terms of cells, we talked about 3 basic types. Bacterial cells, which are very simple, no internal organelles outside of the ribosomes. And then we talked about animal cells and plant cells and some of the organelles in there. I would not worry too much about going back and trying to memorize a whole lot of the organelles, but just trying to look at some of the larger groups of organelles and how they all work together to do their job, like the endomembrane system and the genetic information organelles, and just try to get a feel for how they all work together. Okay, so that was Unit 1 of AP Biology, and I hope that was helpful.
Dropping science like Galileo dropped the orange.